So good afternoon, everyone, and welcome back to the Brainy Boomer Lecture Series. We're so very happy you've all joined us today. In 2007, the McGill University Research Center for Studies in Aging, or the MCSH Education Committee, started the Brainy Boomer Lecture Series in order to suggest practical steps to both improve and maintain brain health, as well as to promote healthy lifestyle choices amongst the most populous generation in history. The MCSH Education Committee, which was founded in 1996, has three main objectives: identifying education needs of healthcare providers, seniors, caregivers, and the public, and to develop responses to meet some of their needs; to enhance the positive image of the aging process by addressing stereotypes and myths about aging; and finally, the dissemination of research on aging. Dr. Natasha Rajah, PhD, is a full professor in the Department of Psychiatry, McGill University, and researcher at the Douglas Research Center. Dr. Rajah received her PhD in experimental psychology from the University of Toronto, St. George's campus in 2003, and did her postdoctoral training in the Helen Wills Neuroscience Institute, University of California at Berkeley. In 2005, she joined the Douglas Research Center, Department of Psychiatry, McGill University. She received a CIHR New Investigator Award and an FRQS Junior 1 and 2 awards in support of her research on the cognitive neuroscience of memory, aging, and dementia prevention. She uses behavioral, neuropsychological, structural, and functional MRI methods to investigate how the human brain learns and remembers past events in rich detail, episodic memory, and how risk factors for late-onset Alzheimer's disease, such as age, sex, and having an apolipoprotein E4 allele impact the structural and functional networks related to episodic memory. Her work to date has helped advance our understanding of the neural basis of age-related memory decline and how the aging brain compensates for these declines. In addition, Dr. Rajah's work has brought attention to the importance of midlife as a critical period in adult development when episodic memory decline first arises. In 2020, she was named the CIHR Chair for Sex and Gender Research in Neuroscience, Mental Health, and Addiction, and was funded to conduct an adult lifespan MRI study to understand why more women are diagnosed with late-onset Alzheimer's disease than men. This work will reveal whether there are sex differences in healthy and pathological brain aging, and if menopause affects memory-related brain function in middle-aged women with risk factors for Alzheimer's disease. Before continuing, we would like to remind you to please mute your microphone on Zoom, and that if you have any questions, you could write them down in the chat box, or you could wait until the end of the conference to ask them live. And now I'd like to invite Dr. Rajah to start her presentation, Why Sex and Gender Matter When Studying Brain Aging and Alzheimer's Disease Prevention.
Thanks, Caitlin, and thank you everyone for attending this virtual talk. Today, I'm going to give you a brief overview of the literature and then what has been happening in my lab so far. But before I start, I always like to start with an acknowledgement slide to thank my team, which is a bunch of graduate students and staff. Here we are after COVID lockdown was restricted, restrictions were removed, meeting in my backyard to celebrate 2021 and all the activities we've been doing in the lab in relation to the research I'm going to talk to you today about. I also want to acknowledge my funding agencies and my collaborators on the projects I'll be presenting to you today. So a brief overview of what I'll be discussing. First, I'm basically going to refamiliarize all of you with the background on Alzheimer's disease, what are the symptoms, and then also maybe some new information to you about the prevalence and incidence of by sex and gender in Canada. And then I'm going to talk to you about why that's important to consider and how do we define sex and gender in our research projects. And after that, I'll go into the type of research that's going on in my lab and present a study that's already been published, which explored sex differences in the effect of age on memory-related brain function across the adult lifespan. And then our current ongoing study that Caitlin alluded to that was funded starting 2020 and is funded for 4 to 5 years. Which looks at the importance of studying brain health at midlife and in women. So just a little background, I'm sure this is a literature review for all of you. So as we know, late onset sporadic Alzheimer's disease is the most common form of dementia diagnosed. It's a progressive, irreversible neurodegenerative disease that causes memory loss, changes in higher order cognitive control abilities, such as judgment, reasoning, strategic organization of behavior. It could also cause changes in mood and behavior, including increases in impulsiveness, difficulties in language, and difficulty in performing activities of daily living, such as driving, taking care of your finances, cooking, personal hygiene. So it's a very disruptive disease. It affects many of our families and is really causing a lot of distress in our society, especially in our advanced aging cohorts. And so one of the hallmarks of this disease are, of course, beta amyloid plaques and neurofibrillary tangles and the neurodegeneration that they cause. But my research focuses a lot more on the cognitive and functional abilities, so especially in the memory domain. So the particular types of memory that are affected by Alzheimer's disease include what's called episodic memory, which Caitlin already kind of defined for you. And that's the kind of area that I conduct my research in. So episodic memory is our memory for our past experiences, but notably in rich contextual detail. So this is our ability to remember what we ate for dinner yesterday, what time we ate dinner, with whom, where we were seated, who was seated beside us. So it really is very detailed recollections of our past experiences. And this is the type of memory that a lot of adults first feel some changes in. And the problem with studying episodic memory is that episodic memory decline is also apparent in what we consider normative aging. So it is completely normal to experience some episodic memory loss. But the question my research tries to address is what type of memory loss in episodic memory may be an early sign of Alzheimer's disease, because we know that deficits in episodic memory are a consistent and early sign that occurs many years before the disease onset. So here's some data from the Public Health Canada that shows you the prevalence of dementia in Canada currently. So in Canada, more than 400,000 adults aged 65 and older are living with dementia, and 1 in 4 adults over the age of 85 have a diagnosis of dementia, of which Alzheimer's disease is the most common. So approximately 76,000 adults are diagnosed with dementia annually, and this will increase as the average age of our population increases, highlighting the importance of really understanding and identifying the earliest time at which dementia might be arising, particularly of the Alzheimer's type, so that we could intervene as early as possible. Now, dementia rates are pretty low among adults between the age of 65 and 69, but they increase with age. And what's important is the prevalence more than doubles every 5 years among seniors, with more than 25% in having Alzheimer's disease at 85 and older. Now, there's been a lot of attention recently to whether or not there are sex and gender differences in the prevalence and incidence of Alzheimer's disease. And this might seem a new thing, but this has actually been going on for a while. So this is a study done back in 1972, where they already noticed that there are sex differences in the prevalence of different types of dementias, with senile dementia, which is now what we call Alzheimer's disease, being more prevalent in females, and vascular types being more prevalent in males. So of the AD cases in Canada, 2/3 are in women. This information may seem new, but as I noted above, the field has been aware of the sex difference in the prevalence since the early 70s. Why this has not received attention in clinical trials or in research until more recently is likely due to a general bias in the medical field that is not focused as much on women's health and sex and gender disparities in diseases. This issue is now being brought into light through advocacy of women and underrepresented groups, and aims to center our understanding of how sex, gender, and diversity in individuals' life experiences may interact with diseases, such as Alzheimer's disease. So here's data from Public Health Canada that indicates that the incidence of dementia, which is the occurrence of new cases in a disease in an at-risk population over a period of time, is pretty consistent. So this is the new diagnosis of individuals in at-risk people. However, the age standardized prevalence of dementia has consistently been more in women and continues to grow over time. So why might this be? Well, the prevalence is the proportion of people in the population who have the disease at a specific time. So factors other than the increase in new diagnoses contribute to it, such as the fact that Canadians with dementia now live longer. So overall, women are disproportionately affected by Alzheimer's disease because they have a higher prevalence in our communities, but also they're also the most likely caregivers of those diagnosed with Alzheimer's disease. So this really highlights the need to study the influence of the role of sex and gender and how that plays in both normative aging, in caregivers, and in individuals with Alzheimer's disease. So this has been one of the focuses of the research in my lab. So I just want to give you a bit of background about what kind of research goes on in my lab. My lab focuses on brain aging and episodic memory function, which I already defined to you. So we focus on episodic memory deficits because it's one of the most consistent and early signs of Alzheimer's disease. We conduct behavioral, neuropsychological, blood marker, genetic, and brain imaging methods in my lab to study brain aging and episodic memory function. And we study adults with and without risk factors for Alzheimer's disease, such as having a family history or having an ApoE4 genotype. We use a variety of methods to try and understand how the brain functions and how it differs across the lifespan in adults with and without Alzheimer's disease risk factors. And this allows us to directly see the functional integrity of memory systems in vivo in adults. We also study both women and men, and we conduct research to look at sex and gender-based effects to detect if there are differences in the way that women and men's brains perform memory tasks as they age. This allows us to determine if the reasons for age-related declines in memory are the same, and if they are not, this is very important to know, since this helps us design therapies and track outcomes with greater precision for care of women and men. Also, we have recently started focusing on early midlife because we know that prevention may be the most effective way to decrease the number of Alzheimer's cases in society. And so we're focusing on adults at midlife, so we may intervene early and provide them with treatments or lifestyle options that might benefit their long-term aging process. So when we started doing research on sex and gender in my lab, we first had to define these concepts. So it's very important to explore sex differences in brain health and aging. But before we do so, it's important to understand what sex and gender may be defined as. So we know that sex and gender, sex is a biological term that people usually collect in biomedical research. So adults often are allowed to check off, are you male or female when they participate in research? And this assumes that what you're reporting is the sex assigned at your birth. But we know that we are more than just sex as a biological variable. We also have to consider gender. And gender is more of a social construct. And it's not just about our gender identity. So we could have individuals that were born as identified as female as birth, that then identify as male as a gender. And so those individuals, we have both questionnaires for their sex assigned at birth and their gender identity. But gender goes beyond gender identity. It also includes gendered lifestyle variables, such as occupational attainment, education levels, the exposure to different types of stressors across one's life, and experiences of different types of diseases across one's life based on gendered life experiences. So it's important to look at both sex and gender in our research, and it's important to explore sex and gender differences in brain health and aging, because we want to know, is the neural basis of cognitive decline the same in both women and men? And do different cohorts implement different compensatory mechanisms to support cognition with advanced age? Does reserve or different lifestyle experiences that promote cognitive health benefit neurocognitive function in similar ways between groups? And do some groups exhibit greater resistance or resilience to aging and neuropathology? So understanding how sex and gender differences in brain health and aging in adults with and without Alzheimer's disease risk factors is very important for our goal of precision health, with its early detection, prevention, delay and or cure for Alzheimer's disease. So in our lab, we've been conducting one study called the Montreal Adult Lifespan Study that spanned between 2012 and 2018. The goal of the study was to conduct a cross-sectional task fMRI study of episodic memory in adults between the age of 19 to 76, who were scanned at both encoding, which is the time when you're asked to memorize a bunch of stimuli, and at retrieval, which is the time in which you're asked to remember things you just memorized. And the goals of this study were first to identify the neural correlates of memory decline across the adult lifespan as a whole. But then a secondary goal, which was led by my student, Savanya Subramaniapillai, was to explore if there were sex differences in age and performance effects or memory effects in brain activation during encoding and retrieval. So the main question of this project was, is the underlying neurocognitive dysfunction associated with age-related memory decline the same in women and men? So to do this, we recruited 141 adults between the age of 19 and 76. They came into our lab on 2 visits. On the first visit, they did neuropsychological assessments, and then they donated their blood for genotyping. And they also had their blood pressure assessed and their glucose levels of their blood assessed to make sure that they weren't pre-diabetic and had uncontrolled hypertension. So after they were screened in from session 1, they came in to participate in an fMRI study. In this fMRI study, we conducted what's called an event-related fMRI study for episodic memory. And what that means is that we're able to collect a whole brain volume of brain activation every 2 seconds. So while you're in the scanner doing a memory task, every 2 seconds, we're getting an image of how active your brain is for specific mnemonic processes. So when you're trying to memorize, when you successfully memorize, when you fail to memorize, etc. So it's a very powerful technique to see in vivo how the brain is functioning when you're doing a memory task. So using event-related fMRI, we had adults lie in the scanner and do a spatial context memory task. And this is a memory task to remember not only faces, but a face in a particular location. So during the task at encoding, so when you're asked to memorize, adults were presented with age variant black and white faces. So pictures of children, adults, older adults were all scrambled together and counterbalanced. They're presented to the left or the right of the computer screen. And adults were asked to encode these faces and the location that they were presented in, so the left or the right of the computer screen. At the same time, to make sure people weren't falling asleep in the scanner, we asked them to rate how pleasant the face was that they saw as well. It was pleasant or neutral. There are 2 versions of the task, an easy task where you were only asked to memorize 6 faces and its location, or a hard task where you were asked to memorize 12 faces and their location. So after memorization, you had a distraction task where you saw 2 words and were asked to alphabetize them. And this was basically to prevent you from rehearsing the faces in your memory and really distract you so we could test your retrieval or your long-term memory ability for these faces. So at retrieval, you saw 2 faces. Both faces were from the previous list, so they were both familiar to you. But what you had to report was which of these 2 faces was originally on the left of the screen or on the right of the screen. So we really were trying to tap into the ability to recollect a face location association in this task. So people did this task for many runs in the scanner, and we collected data for both their accuracy, so how well they remembered the faces and the location that they saw, and their reaction time, which is how fast they made those decisions. So here's the behavioral results, which shows the accuracy for the easy task and the accuracy for the hard task, so how well they did on the task across age, so from 20 all the way up to 80. And what you see is men in pink and women in turquoise. And we did not see any sex differences in performance. And that's very important to take home. So women and men are showing similar patterns of age-related decline in episodic memory for both the easy task and the hard task. And we ran statistics to show that there was an effect of age on memory performance, and there was an effect of task difficulty. The harder task was harder for everybody, but there was no effect of sex on, or an interaction of sex and age on the behavioral data from the study. So then we looked at the imaging study. And to look at the imaging study, we used a multivariate partial least squares analysis technique, which is a very complex mathematical method to look concurrently how behavior and age correlate with brain activation. So remember, people were imaged while they were in the scanner doing encoding and retrieval. And what we wanted to see in our analysis was, how did the brain behave in relation to performance, so how well you did in the scanner, and also in relation to your age. And this allowed us to understand what of the brain activation differences were related to age, independent of performance, and also related to age that impacted performance. And so it allowed us to look at both age and performance effects in memory-related brain function. So here's the main finding from this study. And here you find the brain activation pattern that we saw in the study, the main one. And you see brain activation pattern across global brain regions. So the whole brain is involved in memory tasks. The medial temporal lobe is very important for encoding and retrieval, but so is the frontal and parietal cortices, and they really help direct and control the strategies that you use at encoding and retrieval. So here you have frontal and parietal areas, and you also have some midline brain activation patterns, and you have some medial temporal lobe areas activated as well. And here you have the correlation profiles. So these are correlations that show how this brain activation relates to performance in the scanner and one's age. And what I want you to note is that in both women and men, the performance effects are pretty similar at encoding, and there are some differences at retrieval. So it's encoding, encoding, retrieval, retrieval. And on top, you see the effect of age. And what I want you to notice is only women show an effect of age in this pattern of activation. So with increasing age, you see more activation in these brain regions as a function of performance. So women, older women, activate these brain regions more than younger women, and it helped them successfully encode the face location association on the most hard task. Unfortunately, older women also engaged these regions during retrieval, and that was actually detrimental to performance. So older women tend to engage these regions successfully at encoding, but to their detriment at retrieval. In men, the effects for performance were similar to women at encoding, but this pattern of activation did not vary for age. So here we see a sex-specific effect of age on memory-related brain function. We identified other patterns of activation in this study as well, and I don't want to go into detail here with you, but I want you to take home from both these pictures. So here's another brain activation pattern we identified, and another. What I want you to take home is that the age effects up here and the performance effects are quite different. So men and women are always going in the opposite direction when it comes to age. But when it comes to performance, they sometimes go in the same direction. So what this told us was that performance-related activation, so the way that men and women engage the brain to do these memory tasks, are very similar. And this is not surprising, since we did not see performance effects. Men and women performed just as well as each other on those tasks, but both of them showed age effects, so age-related declines on this task. And what our brain imaging results really highlight is that the reason for that age effect differed in women and men. So the effect of age on memory-related brain function differed. So we identified areas of brain that related to poorer memory function in women and those that were related to poorer memory function in men. This is a very important observation because it tells us that the reason women and men may be performing poorer on these memory tasks in later life are not due to the same neurocognitive changes. So this indicates that perhaps different types of interventions and different types of strategies need to be used to support memory function in women compared to men. So the take-home message of this study is that the neural mechanism underlying memory decline in healthy aging differ in women and men. And even if you don't see behavioral differences in women and men, the underlying brain mechanism supporting behavior may differ. And so the fact that there is a higher prevalence of AD in women compared to men made it apparent to me and many others in the field that we really need to understand why men and women may differ in the way that they're doing these memory-related tasks. And this first study identified brain mechanisms that differ as a function of age in women and men. Now, based on these results, we're now continuing this work to look at brain health at midlife and in women as they experience menopause. So this is an ongoing study in my lab, which we call the BAM study. And there are many research assistants and also trainees that are contributing to this project. And we aim to refine our understanding of sex differences in brain health by not only looking in older adults who are healthy, but also looking at middle-aged adults who are healthy and have risk factors for Alzheimer's disease. And so the goal of this study is to try and understand menopause and its role on neurocognitive brain function in midlife in women, and also look at sex differences in brain function in midlife in men and women. Now, why are we doing this study? So there is rich literature that has looked at menopause effects on episodic memory. And we know that menopause is related to reductions in centrally circulating 17-beta estradiol. And importantly, estrogen receptors are highly concentrated in two neural systems that are integral for episodic memory, the prefrontal cortex and the medial temporal cortex. So we want to understand, how does the brain of menopausal women reorganize its function to support memory at menopause? And this is important to know, given that women are at greater risk for developing Alzheimer's disease. And it's been proposed that how a woman experiences menopause may determine whether or not she ages successfully in later life. So bringing those two literatures together was very important for us to understand and answer the question, why are women more likely to have Alzheimer's disease in later life? Now, there's been very little research in the field looking at menopause specifically and its effect on episodic memory. One of the studies was conducted by our collaborator, Emily Jacobs, back in 2016. And in this study, she had pre, peri, and postmenopausal women and a control group of men perform a memory task in which they were presented with words, one at a time in front of them, and some of the words repeated. So some of them were new and some of them were repeated. And they looked at the activation patterns of words that were new versus repeated, depending on whether they then remembered them later. So this was what we call the subsequent memory analysis, where you look at activations at memorization or at encoding that predicted successful memory retrieval. And they looked at predictions related to novel and repeated words at encoding. And what they found was postmenopausal women showed significant reductions in episodic in hippocampal activation, but there were no effects on memory performance. So there were these brain mechanisms or brain differences in these postmenopausal women that did not directly relate to performance yet, but were detrimental compared to younger women. Now, importantly, to support performance on the task, what postmenopausal women were doing was they were increasing the connectivity, or how two brain regions talk to each other, to a greater degree compared to pre and perimenopausal women. So here you see measures of left and right hippocampal connectivity, and you see that postmenopausal women, but not pre, peri, or men, were showing heightened connectivity. So the way they interpreted this result was that at postmenopause, women exhibit declines in hippocampal activation during a memory task, but they compensate for this with increased connectivity between the left and right hippocampus. And that's why middle-aged women at postmenopause in this sample did not perform significantly different than pre and perimenopausal women. But in my lab, we asked the question, well, what about women who do exhibit episodic memory dysfunction at postmenopause? Some women do report feeling some brain fog and feeling some cognitive dysfunction. So what about these women? How do their brains look? And how do women, are these also the women that may be at greater risk for Alzheimer's disease? Are they the women that are from families with a history of Alzheimer's disease? Are they the ones that have an ApoE4 genotype? And this was the type of question we wanted to answer in the BAM study. So now, based on a lifespan analysis, which indicated that midlife is a critical period and that there were sex differences in brain function, we decided to run this study, which we refer to as the brain health at midlife and menopause. We want to determine if there are sex differences in memory-related brain function using a larger sample and replicate our previous effects. But more importantly, we wanted to focus on midlife and menopause. We wanted to understand, how does chronological aging and menopausal stage affect brain aging and memory function in women? What biological and lifestyle factors support and or hurt cognitive aging in women at midlife? And what is the role of family history of Alzheimer's disease and having an ApoE4 genotype on brain health and cognition in women and men at midlife? So the goal of this study is to collect 400 adults, 120 without an ApoE4 allele who are women, so young, middle-aged premenopausal and middle-aged postmenopausal women, and then the same cohort with an ApoE4 allele and a family history. And then we also had a control group of men to look at the effect of ApoE4 in general at midlife, and also to look at sex differences in brain aging at midlife as well, and to continue our previous work in that area. So the methods for this study are as follows. So participants first fill out an online inclusion questionnaire for screening, and then eligible participants complete a battery of standardized assessments. And finally, people that pass these standardized assessments are invited to participate in our MRI session. So in this MRI session, we get structural images, what we call resting state fMRI, which is scans of your brain when you're asked to just lie in the scanner with your eyes open. And then another task fMRI session similar to the paradigm that we used before. So again, looking at faces and their location. However, because we're targeting middle-aged adults in this study, we used a more complex study. So here we asked adults to encode a face again, but now we have 4 locations. So encode a face in one of 4 locations. And then after you're asked to encode, you're asked to assess how well you think you did in your encoding task. And then at retrieval, you're presented with one face in the center and asked whether or not this face is new, familiar, but you don't remember the location, or you remember the location, it was in the top left, the bottom left, the top right, the bottom right. And then again, we ask individuals to evaluate how well they think. Now, this type of paradigm allows us to measure 2 types of memory responses. One, which we call source memory, memory for face and correct and location. And the other, which we call source failures, which is an inability to remember the location, but you do correctly remember the face. Now, the reason we want to discriminate against these 2 types of memory responses is that we know that with advanced aging at midlife, we have a difficulty in source memory retrieval, but we maintain our ability to remember the recognized faces. So we maintain source failures at midlife. And what we wanted to determine is whether or not adults at risk of Alzheimer's disease who have a family history or a genotype that puts them at risk for Alzheimer's disease exhibit not only difficulties in source memory, but also deficits in source failure. So they also show some inability to recognize faces to a greater degree. So because we just started this study in 2020, we really don't have a lot of data yet. So what I'm showing you here is preliminary results. And because our sample sizes are so small at the moment, we cannot disaggregate our data based on genotype or family history of Alzheimer's disease. But we were able to explore in this smaller sample size how postmenopausal, premenopausal, and perimenopausal women performed on the source memory task and the source failure measurements. And what we see is that indeed at postmenopause only, so remember some of these premenopausal and perimenopausal are middle age, but at postmenopause midlife, women show a decline in source memory performance and a concurrent increase in their ability to recognize, endorse recognizing, but not endorse, correctly endorsing the location of this task. So this tells us that there is a trade-off that occurs at postmenopause where women are more likely to make errors in location judgments, but are just as good in remembering faces compared to younger women. Now, this is, again, preliminary brain activation data that we collected from the scanner, and it is very complicated, but it uses the same method, this multivariate method, where we look at the correlation between age and performance on brain activation. And again, you see a large pattern of brain activation in our primary result that encompasses the medial temporal lobes, the prefrontal cortex, the parietal cortex, and a lot of midline brain structures. And what I want you to notice from this result is first, the first correlation map is an age effect. And this age effect is similar between premenopausal and postmenopausal women, with postmenopausal women contributing to the effect a bit more at retrieval. And so in general, both pre and postmenopausal women at midlife and early on show the same age effect in brain activation pattern. So with increasing age, you see increasing activation in all these brain regions. What's important to note is that how it relates to performance. So in premenopausal women, increased activation in these brain regions supports memory on the harder source memory type of tasks. So more activation here was related to greater success in retrieving the face and its location. In contrast, in postmenopause, greater activation in these regions was related to poorer remembrance of face and location, but it supported better memory for recognition. So what this tells us is that at postmenopause, the brain regions that used to support successful complex memory retrieval, such as face and location in younger age, start to support more simpler processes, such as recognition memory at postmenopause in women. So this is hinting at some effects of menopause on frontal regions and medial temporal lobe regions in relation to source memory in midlife. So this is just the beginning of our study, and we're hoping to now further enroll more and more middle-aged women and also men to continue to look at sex differences in brain aging in this cohort. Now, the critical thing is to enroll individuals who have a family history of Alzheimer's disease and who may be at greater likelihood of having an ApoE4 genotype. So this could really then help us understand whether or not the pattern we see here is being driven by individuals who are at greater risk for Alzheimer's disease compared to those that are not. And that's important to know because it might identify brain mechanisms and cognitive processes that need greater attention at this life stage where intervention might be more beneficial. So in general, what my research to date has shown us is that midlife is a critical period in adult development and maybe when neural correlates of pathological aging emerge, and that there's a need to understand how women and men's brain ages and if having risk factors for Alzheimer's disease has different effects on cognition and brain behavior relationships based on sex and gender. And we also need to center research on women's brain health, so we could develop precision tools for early detection and intervention in Alzheimer's research in women, since 2/3 of the cases of Alzheimer's is in women. And this will improve the quality of women's life in older age. So this is the goal of my research. And if you like what you've heard and you want to contribute your time to this research, I really encourage you to volunteer for the BAM study. We are currently recruiting women and men between the age of 40 and 65. And so I really hope some of you will, reach out to us and want to participate in the study and also encourage your friends and family to participate in the study, because with more participants, remember we only have about 70 participants so far, and our goal is 400 at the end. So we really hope more and more participants between the age of 40 and 65 find this research important and contribute their time. So I know I rushed in some parts, and I'm sorry. So if you have any questions or any details about the logic of the studies or what we aim to do in the future, I'm happy to answer them now. I want to again take the opportunity to thank my students, my funders, and my collaborators, and thank you for your attention. And I hope you found some of what I've reported to you interesting. Thank you.